Thank you. Good afternoon, everyone. Today I'll be presenting this project that we titled Long-Read RNA-seq Demarcates and Trans-Directed Alternative RNA Splicing. So a central challenge in the study of RNA is to understand the effect of genetic elements on the final splicing isoform. One approach to study the effect of genetics is genome-wide studies such as splicing QTL. And this type of study aims to understand the correlation between genotype and the final splicing isoform. So here we have an example of a splicing quantitative loci where the relative expression of different transcripts correlates with the genotype of the QTL. Another approach is to look at allele-specific splicing. That way, we try to correlate the haplotype with the splicing ratios. So, for example, here we have one alternative splicing event where we can see allelic imbalance of the variant located on the cassette exon. So this implies that one of the alleles is preferentially spliced in comparison with the other one. However, the study of allele-specific splicing so far is being only limited to short-read data, thus is very limited to associations between variants that are located very close to the splice site. So here is where long-read RNA sequencing offers a unique opportunity to find the association of variants that locate very far from the splicing event and try to identify which of these genetic elements more significantly contribute to splicing. This is an example of the gene RIPK2. Here we have our long-read RNA-seq data mapped onto this gene. We have a variant located on the middle exon, and we can segregate the reads based on their haplotype. That is the allele that they contain at this variant position. So we'll have our reads in haplotype 1 and haplotype 2 shown over here. At the same time, we have two alternatively spliced exons that are highlighted on these orange boxes. What we can observe is that for this exon, shown over here, all the reads that contain the inclusion of the exon are contained within our haplotype 1. And all the reads that contain the exclusion of the exons are contained in haplotype 2. So the splicing of this exon is strongly linked to the haplotype. Thus, we term this type of splicing event as cis-directed. On the other hand, we have a second alternatively spliced exon where the inclusion and exclusion reads are balanced between the two haplotypes. Therefore, there is no direct correspondence between the haplotype and the splicing. We term this kind of exons as trans-directed. We develop a method to more systematically identify this type of events. So starting from the long-read data, the first step that our method, IsoLaser, performs is variant calling. That is to perform de novo variant calls on the RNA sequencing reads. After identifying variants at the single read level, we then proceed to phase these variants and group the reads into the haplotype that they correspond. And after that, we perform a linkage test. This test aims to find any linkage between the haplotype values of every read and the splicing values. That is, whether every read includes or excludes a given exonic part of interest. Exonic part being the segment of the exon that differentiates different isoforms from each other. The way we calculate this linkage is using the adjusted mutual information. Very briefly, adjusted mutual information aims to understand the relationship between two random variables. That is to say, how much we can learn from one random variable in order to predict a second one. In this case, how much we can learn from the haplotype in order to predict the splicing outcome. We applied our method into the ENCODE dataset that consists of multiple human and mouse samples. That includes multiple human tissues, cell lines, and also a small Alzheimer's disease consortium cohort that contains 5 AD patients and 4 controls. The first thing we did with this data set was to calculate the percent splice in, that is the splicing ratio of the exonic parts of our different samples, and cluster them. And what we can observe when we cluster the PSI, that is the splicing ratio of these different samples, what we notice is that our samples tend to cluster mostly on the tissue that they originate from. So that is to say, samples that derive from the same tissue tend to share similar splicing profile. And that is further quantified using the Milas variation of information. So lower values, that means more correlated, the labeling with the clustering. So that, however, that's being already reported. It's already well known that the splicing is very tissue specific. So different proteins, different RNA binding proteins are expressed in different tissues. So we proceeded to perform this clustering, but instead of using the PSI, we now use our metric, the adjusted mutual information. And what we can see now is that our samples will cluster mostly based on the donor that they originate from instead of the tissue. And once again, this is quantified using the Milas variation of information. So what this is saying is that despite different tissues having their own alternative splicing profile, the linkage between the genetics and the splicing is consistent across different tissues and depending on the donor that they come from. Using this metric, the adjusted mutual information, that's how we can discriminate between our cis-directed and trans-directed events. Here we have a summary of how many cis-directed events we find in our human tissues. The number spans between 100 and 200 approximately. In average, we have 1 cis-directed event per gene, some having multiple. And in terms of the technical specifications, it tends to depend highly on the coverage of the sample and the number of testable heterozygous variants that we find. We then proceed to look at what are the genes that contain these cis-directed splicing events. So we perform a gene ontology analysis, and we summarize the parent terms of the ontologies that are more significantly represented. What we find is that among the different ontology groups, there is an overrepresentation of immune-related functions. So we have 3 terms over here: intracellular signal transduction, innate immune response, cellular response to interleukin-1. All of these parent terms contain multiple ontology terms within them. Overall is the most overrepresented category. So that is to say that genes that have an immune-related functions tend to have a cis-directed splicing mechanism. We then look at more specifically into the exonic regions that are spliced in this manner. And what we find is that when we look at the conservation score, so the FASCON score is the conservation across 100 different species of vertebrates. What we find is that the exonic parts that are transdirected, that is not associated with the genetics, tend to be more highly conserved than the exonic parts that are cis-directed, the ones correlated with the genetics. And what is more interesting is that depending on the strength of the linkage in this case measured by the delta PSI, that is the PSI difference between the 2 haplotypes. Depending on the strength of the linkage, we see a correlation, a negative correlation with conservation. So those exonic regions that are more strongly correlated with the genotype tend to be less conserved across all the species of vertebrates. Going further into our method, we realize we can further leverage on the allele-specific information that we collect at the sample level. So when we identify haplotype association for individual donors, all we are learning is that there is a correlation between the haplotype and splicing. The next question is then, can we identify the specific genetic variant that is associated with the splicing? So we leverage on the allele-specific signal from multiple donors, and what we do is what we call a narrowed down SAV. SAV stands for splicing associated variant. So through this approach, we try to find the variant that is consistently linked with the splicing event across multiple donors. That is to say, the variant that maintains the linkage, regardless of the haplotype. This method is part of our isolaser package that will leverage on the multiple samples of your cohort. And another advantage is that we can also reinforce association. So that is to say that some samples may not be testable because they either don't have enough read coverage or they have monoallelic expression. So also by combining the data of multiple samples, you can expand the number of testable events, testable SAVs. So it's worth noting at this point that the SAV is a similar idea to the splicing QTL. We are finding variants that are associated with splicing, with the major advantage that because the association is done at the read level, we don't need as many samples in order to get statistical power. As long as we have enough read coverage, we can establish a direct correlation between allele and splicing. We then seek to validate our SAVs that we found on the ENCODE human tissues. And the first thing we did was to overlap our SAVs with the splicing QTLs reported by the GTEx consortium. So in this graph, we looked at the p-values calculated by GTEx of the SQTLs that overlap our SAVs. And we also find a set of match controls. What we find is that our SAVs have more significant p-values compared to our set of match controls, which suggests that we are in fact finding functionally significant variants. Another mode of validation is to see if our SAVs significantly disrupt the binding of RBPs. That is to say, if a variant, if by introducing a new allele disrupts the binding motif of a protein, then that suggests that it's functionally significant. And by using DeepRipe, which is a deep learning method that calculates the effect of allele on the binding of given sequences, we find that our SAVs more significantly disrupt the binding of RBPs. Once again, suggesting they are functionally enriched. Looking into our Alzheimer's disease cohort, we performed this analysis when we merge our AD donors and our healthy donors. And one of the SAVs that we find, this shown over here, has a significant linkage with splicing event, shown over here. What we found that is very interesting is this condition-dependent signal, where even though the allele, which is shown on the side, even though the allele of this variant is strongly linked with the splicing, the direction of this linkage is inverted depending on the condition. That is to say that in AD samples, the T allele is linked with inclusion, whereas in the healthy donors, the T allele is more strongly linked with the exclusion of that exonic part. And this splicing event, of course, is very significant because the disruption of, or the exclusion of this region disrupts one of the ubiquitin domains of the ubiquitin C gene. And the disruption of these ubiquitin-like domains disrupt the ubiquitin-free pool within the cells that is involved in protein degradation. So to conclude, we developed a pipeline that allows for the classification of different modes of splicing regulation. Cis-directed events maintain similar PSI regardless of the tissue of origin. Immune-related genes are highly enriched in cis-directed regulation. Cis-directed exonic parts are lowly conserved among vertebrates. And isolaser joint analysis allows for the discovery of splicing-associated variants in small cohorts. Lastly, I would just like to acknowledge my lab, the Shao lab at UCLA, especially my advisor, Grace Shao, and my colleague Kofi Amoa, who's a co-author in this paper, funding sources, and all of you for listening. Thank you.
Maybe there's time for a very quick question.
Other questions? That's fine. Any questions? I have a question.
Beautiful work. So you mentioned that conservation difference between the Cs acting. Why do you think that is? Why do you think they're not as much conserved?
Yeah, that is short answer is I don't know. There is being a body of evidence that says that allele-specific regulation, be it splicing or expression, is sort of an evolutionary novel mode of regulation, meaning that the target genes or exons are less conserved. So that is a pattern that's been reported in the past. The explanation is still not well established, I would say. We can speculate that it's an evolutionary novel mode of introducing diversity. But as far as I'm concerned, we don't have still a clear answer to that.
And more quick question: are you able to do that from both nanopore data and PacBio?
Yeah, we've used all of the results I've shown here are from PacBio, but we are able to run this pipeline on nanopore as well. Yeah.
Thank you.
Yeah.